Hey guys, we're going to learn about meiosis really quickly. There's two types of cell division. There's mitosis and meiosis, and we're going to concentrate first on meiosis. You'll also hear it called meiosis, so don't get confused with that. What's important to understand is we're making sex cells here. So they're your gametes, that's another vocab word. This would be your sperm and your egg. And what's the whole purpose of doing this? Why do we need to make egg and sperm? Well, the whole point is for reproduction. And the point of reproduction is to continue a species. So that's what we want to focus on here. So I kind of want to show you a trick to memorize this, and it's really fun, really simple. So anytime your hands are apart, it's a haploid. Anytime you're together, it's a diploid. Now you should have learned those in your biology class, but here's just a brief review. A haploid, there's only one copy of the genetic information from one parent. Diploid, there's two copies of the same genetic information. So you can see there's two number 3s, one from mom, one from dad. So you have egg and sperm, your hands are apart, so that's a haploid. When they come together for fertilization, that's now a diploid because your hands are together. S phase, they're going to replicate as chromosomes. Now you're going to undergo meiosis 1 where the chromosomes are split apart. That's now a haploid because your hands are apart. Meiosis 2, sister chromatids are going to separate. And you're still a haploid. So you're going to make 4 sex cells, and they're all haploids. So we're going to kind of show this here. So you have sperm and you have eggs. They each have 4 chromosomes. You can see them labeled. The way that I like to teach this, it's the same person with the same genetic information, but each chromosome carries different information. So number 1 may have your eye color, number 4 may code for height. It's the same as this would be like your biology notes, your physics notes, chemistry notes, earth science notes. It's all science, but it's different subjects. So that's a really easy way to think about it. So you have 2 haploid cells because you only have one person's genetic information for each chromosome. Then fertilization happens. And now you have a diploid. This is a zygote, or this is a fertilized egg, a baby. Wait for the bell. Now it's a diploid. So the first thing that happens is interphase. Interphase has 3 steps, G1, S, and G2. G1, it grows a little bit. S phase, your chromosomes replicate to get an identical copy. And that's what you're used to seeing on the board when your teacher draws it. So we're just going to quickly replicate these chromosomes. And I'm still a diploid. Okay, all that you're doing right now is this. You're just replicating them. G2, the cells grow again. Now we're going to go into prophase 1. And prophase 1 is really cool. It's my favorite step. In prophase 1, your homologous pairs are going to snap together in synapsis. So they're literally going to snap together. Now they're called homologous chromosomes because they carry the same genetic information. So mom's number 4 pairs up with dad's number 4. And something really cool can happen when they get this close. Some of dad's information can go on mom's chromosome, and some of mom's chromosome can go on dad's chromosome. And that's called crossing over. And that's one way we get genetic variation. So for instance, this chromosome would have made me. I look a lot like my dad, and then I have a little bit of my mom, so I have her eye shape and eye color. So that would represent me, and that's how you get genetic variation within families. So we're going to make the rest of these snap together through synapsis. Now another vocab term is homologous pairs. Homo in science means same, so that's why we call them homologous. Another name is tetrad because there's 4 pieces, so tetra means 4. So now that prophase 1 is done, we go into metaphase 1. Metaphase 1, your homologous pairs line up along the equatorial plate. And what's really important to realize is just because all of the blue ones, the first 3 blue ones are over here, doesn't mean that the 4th one has to have blue over there. That's called random alignment or random assortment. All right, so now we're in metaphase 1. Meta is for middle. Now, anaphase, I like to teach my kids, anaphase starts with an A, so anaphase means apart. Homologous pairs are going to be pulled apart and go towards the opposite ends of the pole. Once anaphase 1 is complete, I'm starting to become a haploid. So what we're doing is this part of the dance, the finger dance. So then telophase is going to happen, so the cleavage furrow starts to form, and then cytokinesis, you get 2 completely different cells. But we're not done yet because we don't have 4 cells yet. We only have 2, so we're going to go right back into prophase 2. Now we don't have an interphase because you're already replicated, so there's no need to get that. So we're going to go into prophase 2. Prophase 2, the nuclear membrane is going to break down again. Then metaphase 2, the chromosomes line up in the middle. Anaphase 2, this time sister chromatids pull apart. And then in telophase 2, you'll have the cleavage furrow form. And in cytokinesis, you get complete cytoplasm division. So now I have 4 completely unique cells due to crossing over. And I'm haploids. So here's the finger dance again. Mom's egg, dad's sperm. They're haploids. They come together as diploids. And an easy way to remember that actually forms a D. So diploid, then S phase, they replicate. Then meiosis 1, homologous pairs separate. They're haploids. Meiosis 2, sister chromatids separate. Meiosis 2, here's 2 fingers. I now have 4 cells. They're all haploids. And that's meiosis.